Hello everyone. Today we explain the lab 9, which is about the acid-fast stain. It also called Ziehl-Neelsen stain. It is the most important differential stain used in microbiology lab because it allows to identify the most important medically important bacteria such as Mycobacterium tuberculosis, which cannot be stained by using the ordinary stain like a gram stain. The acid-fast stain is both differential and combined staining technique. Why it is differential stain? Because it differentiates between two groups of the bacteria, those that are acid-fast and those that are non-acid-fast. The organism we focus on this lab include members of the genus Mycobacteria, especially Mycobacterium tuberculosis, as well as certain species of Nocardia. To understand why these bacteria are acid-fast, we need to understand the structure of cell wall of this microorganism. These microorganisms have cell walls that contain large amounts of mycolic acid along with other complex lipids, fatty acids, and waxes. This thick, waxy, and hydrophobic layer make them very resistant to the ordinary staining method like gram stain, and even to many chemical disinfectants. Because of this unusual cell wall composition, a special staining method, like acid-fast stain, is required to visualize these microorganisms under the microscope. In the acid-fast stain, we use 3 different chemicals because we use more than one reagent. This method is also called combined stain. The first one is carbolfoxime. This is the primary stain. It's red color that enters the cell with the help of the heat. The second one is acid alcohol. This is the decolorizer. It removes the red color from the non-acid fast bacteria but cannot remove it from the acid fast one. The counter stain is methylene blue. It's a blue that colors the non-acid fast bacteria. So we can differentiate between the two groups of the bacteria, acid-fast and non-acid-fast, by using this stain. So the mechanism of the acid-fast stain. When we add the primary stain, carbolfoxime, that contains phenol red, with the help of the heat, this stain can dissolve the lipid in the acid-fast bacteria and enter the thick, waxy cell wall of the acid-fast bacteria. After washing the smear with a strong acid alcohol decolorizer, this decolorizer removes the red stain from all non-acid fast cells, while the acid fast keeps the red stain because the waxy cell wall does not allow it to wash out. And finally, the cell that lost the red stain, it takes up the counter stain, which is blue. Therefore, we can visualize the difference between the microorganism clearly under the microscope. Why it is called acid-fast? Because once the acid-fast bacteria absorb the carbolfoxime, they cannot be easily decolorized by acid alcohol. That's why it's called acid-fast. What about the procedure? I will also provide a video that shows the procedure. First, we should prepare the smear. As you know, we make the smear by spreading the bacteria on a clean slide, then air dry, then heat fixation. After that, we put the slide on a hot plate and cover it with the carbolfoxime and heat it for about 5 minutes. Then we will wash with the water. The next step is the decolorization by adding the acid alcohol drop by drop for about 30 seconds. It will remove the stain from all non-acid fast cells. Then we will wash with water. The next step is the counter stain. We put the methylene blue for about 1 minute. Then we will wash. After that, we will dry the slide and then observe it under the microscope. The procedure of the acid-fast stain. After I prepare the smear,
You know how to prepare the smear. It's spreading the bacteria on a clean slide, allowing to the air dry, then fixation, and allowing to be cold. Then I will do the procedure of the acid-fast stain. The primary stain is carboxyl. Then we add the acid alcohol drop by drop for about 30 seconds, and a counter stain is methylene blue. First of all, I put the beaker on a hot plate because I need the steam before I add the first stain, primary stain. Then I will add the primary stain, carboxyl, for about 5 minutes. What are the principles of this stain? This stain, by the help of the heat, it enters in the cell wall of the Mycobacterium tuberculosis. We don't allow the stain to be dried. If the stain is dried, we add more stain. So you know the bacteria is colorless. We add the red stain. Both types of the bacteria will take the red stain. After 5 minutes, remove the stain and allow it to be cold. Then you will wash the slides. Until the color will be disappear. Next step is add the acid alcohol drop by drop for about 30 seconds. This stain will remove the red color from all non-acid fast bacteria, while the acid fast bacteria will retain the primary stain. I wash until the color will be disappear. The counter stain is a methylene blue. Add to the slide for about 1 minute. The non-acid fast bacteria will take the counter stain, which is blue, while the acid fast bacteria, by the help of the heat, it will take the primary stain, which is carboxyl, and retain this stain. Will not be removed during the decolorization. It retains the primary stain and appear red under the microscope, while the non-acid fast bacteria will take the counter stain after the step of the decolorization. After 1 minute, wash the slides until the slide there are no more stain on the slide. Then we dry the slide and examine under the microscope. And I will available the slide that show you the acid fast stain and non-acid fast stain.
Reading the acid-fast stain result. The acid-fast bacteria such as Mycobacterium tuberculosis, it keep the primary red stain, so it appear bright red or pink under the microscope, while the non-acid-fast bacteria cannot hold the red stain, so it take the blue counter stain instead and appear blue along with the background. In the microscopic image, you clearly see the red acid-fast bacteria, which is bacilli in shape, visible against the blue background. This image, it's the outcome of the Ziehl-Neelsen stain, which is showing that the acid-fast bacilli are visible as a red to pink root that keep the carboxyl stain because of their waxy cell wall, while the surround or the background with a non-acid-fast organism is stained blue, which is take the methylene blue color, the counter stain. This rapid result is essential for identification the pathogenic like Mycobacterium tuberculosis in a sputum smear. Here in this slide, we will summarize the entire process and the outcome of the acid-fast stain. As you know, bacteria is colorless. First, we add the carboxyl, the primary stain. The both type of the bacteria will staining purple. After that, when we add the acid alcohol, the decolorizer, the acid-fast bacteria retain the primary stain because they have a thick cell wall, waxy cell wall that prevent the decolorizer from washing it up, while the non-acid-fast bacteria will lose the primary stain and become clear. After that, add the counter stain methylene blue. The acid-fast bacteria, it retain the original stain, primary stain red color, while the non-acid-fast bacteria take up the new stain and appear blue. And at the end, you should know this staining technique is very important in diagnosis disease like tuberculosis, and understanding this mechanism is very important for your professional training. And thank you.